Fine structure of HIV. Fine structure of HIV has been studied by the help of X-ray diffraction and spectroscopy. HIV looks like rounded structure measuring about 120 nm in diameter, found on the surface of macrophages, B cells and CD4 cells. A number of viruses are seen budding from the cell surface of CD4 cells or macrophages, while the rounded mature viruses called capsids are ready to leave the cell, which are found attached with the antigen receptors on B cells or macrophages. Virus is covered by rounded protein coat. The outermost protein consists of glycoprotein 120. Beneath outermost protein layer is GP glycoprotein 41 transmembrane protein layer, which crosses phospholipids bilayer of the capsid. The GP120 protein coats are derived from the membrane of host cell that contains host cell surface receptors such as HLA class 1 and HLA class 2 molecules. Below GP41 and GP17 and GP24 protein layers enclosing the HIV genome. HIV genome is composed of two single-stranded RNA molecules tightly bound to nucleocapsid proteins P7. Each RNA strand is associated with enzyme reverse transcriptase and also with enzyme needed for the development of virions such as proteases, ribonuclease and integrases. The matrix made up of protein P17 surround the capsid. The end of each HIV RNA possesses additional RNA nucleotide sequence called long terminal repeats, which act as switches to control production of new virions. HIV genome contains 9,000 to 10,000 nucleotides. The genome is characterized by the presence of long terminal repeats and 3 long terminal repeat nucleotide sequences which act in binding for transcription factors. The genome contains 3 structural genes such as GAG, POL and ENV that contains the information for making structural proteins for new virion. The GAG genes encode for synthesis of core proteins GP24 and P11. The POL gene encodes for 2 reverse transcriptase enzymes, one P10 protease enzyme and integrase enzyme. The ENV encode for 2 outer glycoprotein enzymes GP41 and GP120. HIV-1 genome also contains 3 gene fragments called as regulatory genes, such TAT genes, TAT gene, which is called as master switch. It encodes protein for attachment with viral RNA. It can also accelerate transcription. NEF gene, which downregulates host cell HLA class 1 molecule and CD4 cell. And REV gene mediate use of mRNA of host cell. Other genes are VIF, VPR and VPU. VIF gene monitors the infectivity of free and not cell-bound HIV. VPR gene acts as a weak transcriptional activator, while VPU is required for the efficient building of new virions from CD4 cells. Inside the host, both the LTRs are responsible for regulation of viral expression. LTRs contain regulatory component associated with gene expression. For instance, the TATA promoter contains polyadenylation signal sequences, the transcription factor NF-alpha beta and SB1 binding site, the transacting response site where TAT proteins bind negative regulatory element, whose deletion accelerates the gene expression. It has been observed that HIV-1 and HIV-2 differ from each other. VPR gene is found in HIV-2 but not in HIV-1. With above feature, the genome of HIV is able to program its selfish gene terrorizing the human population. Some proteins from host cells are embedded in envelope of virus and approximately 70 copies of complex HIV protein protrude through the rough surface of virion. The protein code GP120 is envisioned as a potent vaccine molecule against HIV infection. Life cycle. The life cycle of HIV include mode of infection, docking and tropism, reverse transcription for DNA synthesis in the host, and replication cycle for emergence of new virions for DNA synthesis and emergence of new virions. Mode of infection. Transmission. Epidemiologically, AIDS disease occurs throughout the world. HIV-1 dominates in worldwide while HIV-2 predominates in West Africa. HIV is passed from one person to the other when infected blood, semen or vaginal secretion come in contact within uninfected persons, broken skin or mucous membrane. AIDS affects men and women equally. Many women get AIDS disease from their husband who have multiple sex with many HIV positive women. AIDS virus is transmitted by sexual contacts, which may be of following kinds. Penile. The penile mode of transmission is most common method of HIV transmission. Penile vaginal transmission. Penile vaginal heterosexual intercourse is also a common mode of HIV transmission. If in males, HIV infected individuals, especially suffering from urethritis and epididymitis, reveal inflammatory lymphocytes and monocytes. Oral sex has been observed to be less efficient means of HIV transmission. HIV transmission through blood and blood products, contaminated blood transfusion, syringes and needle transmit AIDS virus. Now disposable syringes and regulatory laws regarding blood collection storage reduce the risk of HIV infection. Transmission of HIV in occupational and health workers dealing with collection of blood samples working with AIDS virus involve risk of HIV infection. Mother to child transmission route. The infection of HIV from mother to child can occur in uterus during pregnancy. At the time of childbirth, AIDS virus can be transmitted from HIV infected mother to her infant child. Transmission of HIV by other body fluids. According to WHO, there is no evidence that kissing or saliva exchange involve risk of HIV infection in men or women. Docking and tropism. After transmission, the AIDS virus enters into CD4 cells or macrophages and B cells. The outer protein coat of GP120 of the AIDS virus is susceptible to the antigen receptor of CD4 cells. High affinity of interaction GP120 of HIV virus and antigen receptor of CD4 cells. The CD4 cell receptor is called CXCR4 and receptor of monocyte and macrophages are called CCR5, which is predominant in EG with chemokine receptors and microglial cell stage of disease. The T-tropic isolate or Syncytia including strains replicate in CD4 cells as well as macrophage using alpha chemokine receptor CXCR4 for entry. The alpha chemokine SDF1, a ligand for CXRL1, suppresses replication of T-tropic HIV1 isolate proteins, which isolates persons with fine CCR5 receptors of macrophages are more susceptible to HIV infection, and the process is called as docking. After a series of interactions, surface coat of HIV merges with plasma membrane of CD4 cell. The RNA strands and enzyme reverse transcriptase flows into the cytoplasm of the CD4 cells or macrophages. Reverse transcription for DNA synthesis in host and replication cycle for production of new virions. After docking and entrance of viral capsid into host cell, the enzyme reverse transcriptase initiates its action by liberating RNA strand from attached protein. It utilizes viral RNA nucleotides that are degraded by enzyme ribonuclease to synthesize DNA complementary to viral RNA nucleotide. Except for that, in the newly synthesized single-stranded DNA, thymine is incorporated instead of uracil For several years, the central dogma of molecular biology, DNA to RNA to protein, was considered to be the only molecular mechanism to occur. However, Howard Tamin discovered that in retroviruses such as HIV or HTLV, RNA reverse transcribes RNA into DNA mediated by enzyme reverse transcriptase. Howard Tamin was awarded Nobel Prize for his outstanding discovery in 1975. The working of enzyme reverse transcriptase can be compared to untrained computer assistant who translate German language in English and makes lots of mistakes. Enzyme reverse transcriptase transcribe RNA strands very hurriedly. The faster it transcribes, more mistakes it incorporates. These mistakes are referred to as mutations. Thus, during infection and viral proliferation, various virions are produced. Enzyme RT transcribed reverse transcriptase transcribes some additional sequences called long terminal repeats LTR at both ends of the DNA strand. The enzyme RT disintegrates after synthesizing new single-stranded DNA. The LTRs direct the synthesis of complementary DNA strands called as cDNA. The two newly synthesized DNA strands assume double-stranded DNA like that of host DNA. The newly synthesized double-stranded DNA moves from cytoplasm to nucleoplasm, where it integrates into the host chromosomal DNA mediated by the enzyme integrase. LTR of provirus destroys nucleoplasm and also hosts RNA machinery, creating syncytial condition. The HIV viral double-stranded DNA is called as proviral genes virions. Thus, the integrated viral DNA may lie in dormant stage. The viral DNA becomes activated by certain cellular transcription factor such as NF-kappa B, that is upregulated from activated CD4 cell. The viral integrated cDNA starts transcribing mRNAs. The smaller piece of mRNA produces regulatory protein TAT that induces new virus protection. The REV inhibits splicing of mRNA and producing full-length RNAs that are actually viral genome which code for GAG and ENV proteins. The proviral genes become activated by more accumulation of cDNA molecules in host cell. Cytomegalovirus or Epstein-Barr virus also activate these DNA. Thus, numerous new virions are produced ready to infect new CD4 cells, macrophages and B cells to continue the propagation of their selfish genes. During synthesis of various proteins of new HIV, the virus use host protein translational machinery such as ribosome enzyme and various protein factors. The new virions are terrible creatures causing various cytopathic and clinical changes and eventually demolition and suppression of host immune system resulting into AIDS disease in men. The integrated genes of HIV1 with host finally takes first step of viral cycle to produce new virions. The new virions is produced at plasma membrane of host cell. The ENV polypeptide is carried into endoplasmic reticulum and then transported to Golgi body where it is hydrolyzed into two HIV envelopes GP41 and GP120. These are transported to plasma membrane where GP120 anchors with host cell. The GAG pol polypeptides along with HIV1 genomic 2 RNA strands associated with inner surface membrane. They start budding from new virions which infect other cell. Clinical manifestations of AIDS. The clinical manifestation of AIDS comprises various symptoms due to acute infection and immunodeficiency of various immune components cells such as CD4 cells, B cell, NK cells, macrophages, stem cells, It also defects in antibody synthesis and acquisition of an opportunistic infections. The acute or initial infection of HIV consists of rapid viral replication leading to abundance of HIV in peripheral blood, approaching several million viruses per ml called as acute viremia. Acute viremia is associated with depletion of CD4 cells and activation or an increase of CD8 cells. CD8 cell increases then declines. A good CD8 cell controls viral load. HIV infected cells also induce good antibody protection. A number of CD8 cells are also linked with slowing disease progression and better prognosis, although CD8 cell does not completely eliminate HIV virus. In this condition, in usually 2-4 weeks of HIV infection, most individuals develop influenza-like system and mononucleosis like illness, having fever, lymphoadenopathy, pharyngitis, rashes in mouth, milia malaise, headache, nausea and vomiting. Patients show weight loss, enlarged liver and spleen. Patient becomes susceptible to other infection as a result of various HIV induced immunodeficiency. Immunodeficiency of CD4 cells. The major important immunological reason consists of suppression and destruction of CD4 cell. In AIDS patient, the number of CD4 cell is less than 200 per ml. It has also been observed that CD4 cell depletion occurs in blood, which is proportionally a small fraction of total T cell production and proliferation in lymphoid organ such as in thymus, spleen, and lymph nodes. In AIDS patients, the DNA responsive for generation of T lymphocytes may be affected, including stem cells and deficiency of CD4 cells in bone marrow. The depletion of CD4 cell occurs in various ways. CD4 cell possess receptor for GP120 proteins of AIDS virus. The surface membrane of CD4 cell is destroyed during attachment of virus with CD4 cell membrane. During docking, HLA class second molecules are destroyed so that CD4 cell become ineffective in recognizing and presenting HIV antigens to CD8 cell for destruction. The HIV infected CD4 cell fails to express IL-2 and IFN gamma, leading to significant decline in the functioning of immune component cells. Immunodeficiency and depletion of B cells, suppression and deficiency of macrophages. B lymphocytes are called cells are called as immunocompetent cells as they play important role in combating antigen in blood and tissue. AIDS patients become incapable of inducing sufficient production of IgM antibodies. In HIV infected person, the raised IL-6 indicate B cell abnormality. B cells undergo transformation by Epstein-Barr virus and cytomegalovirus, and they can be infected by HIV-1 as the transformed gene. Genes transcribe CD4 receptor on their surfaces. Macrophages act as a reservoir of HIV-1 and they also play important role at various stages of HIV-1 infection in men. In brain cells, lungs, and lymph nodes, 10 to 15% of macrophages are infected by HIV-1. The Kupffer cells of liver do not reveal HIV-1 infection in CD4 cells. The HIV virions are released, whereas in macrophages, HIV-1 can replicate and they accumulate in large number in vacuole or cytoplasm. Unlike CD4 cells, the HIV virions are not easily released but they aggregate in large numbers in macrophages. Macrophages act as a reservoir of large number of HIV-1 which do not kill macrophages and instead macrophages spread HIV in entire body. The alveolar macrophages causes formation of lung lesion and the glial cells of brain causes neurological problems. The AIDS patient becomes susceptible to secondary infection. Other diseases in opportunistic infection comprise cervical cancer, coccidioidomycosis, cyclospora, diarrheal disease, histoplasmosis, herpes simplex infection, and isopsoriasis. Mycobacterium avium and Mycobacterium tuberculosis causing tuberculosis, Pneumocystic tyranny cause pneumonia. Patients also become infected with Salmonella, Toxoplasma, and Candida albicans, causing typhoid fever, encephalitis, and mouth rashes, respectively. PCD, AIDS and programmed cell death. PCD is a homeostatic physiological suicide mechanism in which cell death occurs during normal tissue formation. During PCD or apoptosis, decrease in cell volume and other profound change occur, such as rupture of plasma membrane and fragmentation of nucleus takes place. DNA breaks into small fragments. The dying cell produces a small membrane bound apoptosis body which are phagocytized and digested. Besides various mechanisms mentioned earlier, the protein code GP120 of infected cell binds with antigen receptor of activated CD4 cells, of which may activated infected CD4 cells, and the uninfected cells are programmed for death directly. Diagnosis and symptoms. The diagnosis of AIDS patients Diagnosis of AIDS patient can be done by detecting antibodies against HIV-1 in serum. Usually after 3 months of infection, antibodies appears in blood. For diagnosis of an individual HIV positive, there are though two simultaneous confirmatory tests: detection of antibodies in serum by ELISA against AIDS virus and counting by fluorocytometry with less than 200 CD4 cells per ml of blood positively confirm HIV infection. Prevention. There is need to form national, state and district level committees to promote health education regarding AIDS disease. Health education should be formulated for all groups of individuals in communities, especially college and school level students. The education should be given regarding mode of infection and especially life cycle and clinical aspects of HIV. The education must be provided to commercial sex workers and the visitors of commercial sex workers regarding precautionary measures and use of condoms. Adolescent and grown up people must be educated how to be good boys and girls and women, good husband and wife. No AIDS vaccine has been developed so far because of three reasons. Firstly, the replicating virion produces mutational virus with always variant GP120. Secondly, it is impossible to develop vaccine against ever changing antigens and their responses for HIV infected cells and cell free virions. Thirdly, in case of AIDS, it is not possible to give vaccine to healthy person to prevent infection. Treatment of AIDS patient involves chemotherapy. One of the most effective drug is called zidovudine, didoxycytidine, and didoxytyrosine, which inhibits reverse transcription to prevent formation of cDNA. The drugs also check emergence of new virions. Thank you.